All right, good morning, everyone. I want to start by thanking the organizers for inviting me to speak about our work and whoever had the idea of sticking me behind Steve Goldstein. So today I'm going to tell you a story very briefly about an ongoing program we have in the lab where we're developing inhibitors and activators of a renal potassium channel, KIR4151, because we think it might be a good target for treating disorders of fluid balance disorders. How do we move forward here? Hang on. Great. So our overarching mission, before I get started, Colin, can you see the screen without your glasses? Okay. This is important. I want you to see this. I want to hear some comment from you. So our overarching mission is translating discoveries into novel therapies targeting ion channels through early stage drug discovery and target validation. As all of you know, drug discovery is a long and expensive process. Academic labs can't do it all by themselves, so it usually requires partnering with a pharmaceutical company. And even drug companies don't have they can't risk the investing into a target that hasn't been thoroughly validated, at least with genetics, but ideally with pharmacological tool compounds. And that's where we come in. We try to develop high-quality small molecule tool compounds for de-risking these targets so that we may entice drug companies to make the rest of the investments. And so over the years, the last 15 years or so, we've developed and published a number of small molecule inhibitors and activators, mostly of the entered rectifier family, simply because they were attractive drug targets. And also the pharmacology when we got into the field was basically barium, micromolar to millimolar concentrations of barium. And so as you can see from this list, we published a number of these, and the ideal model is that we publish them. A drug company like Tocris or Sigma will begin synthesizing them, make them publicly available, commercially available, and people use them and to study their favorite target. And so this is just a sample of some of the papers that have been published over the years, leveraging our tool compounds to investigate their favorite target. So the story I'm going to tell you today is about a KIR channel that's expressed primarily in the kidney. And just as a refresher, the kidneys are two organs in your lower back that play the important role of regulating blood volume and pressure. And they do this by filtering about 140 liters of blood a day through the glomerulus, which creates an ultrafiltrate that enters the nephron. And the ionic composition of this is modified by ion channels and transporters on its way to the bladder where it's lost as urine. And in general, water follows sodium. So sodium chloride reabsorption promotes an increase in extracellular fluid volume and blood pressure, whereas inhibition of sodium chloride reabsorption causes diuresis, lowering of blood pressure. So the region of the nephron that we're concerned with today is called the distal convoluted tubule, which is shown here. Here's a cartoon diagram of the major transport pathways that are present there. Sodium chloride reabsorption in the DCT accounts for about 10% of the total filtered sodium. So it's an important sort of drug target for diuretics, specifically thiazide diuretics, which inhibit the sodium chloride symporter. This is an electroneutral sodium chloride transporter that's regulated essentially by phosphorylation. Sodium that moves into the cell is transported out or pumped out across the basolateral membrane back toward the blood by the sodium pump. Chloride then enters the cell, exits through a conductive pathway through CLC-KB channels. And it turns out that KIR4151 channels play a critical role in this process, in part by recycling potassium across the basolateral membrane to keep the sodium pump energized, but also by regulating the membrane potential, which regulates how much chloride leaves the cell, and consequently, how much chloride is in the cytoplasm. And it turns out that the activity of the sodium chloride cotransport is regulated by phosphorylation via a chloride-sensitive Wnt/SPAC kinase pathway, which is summarized here. So you can see that changes in chloride regulate the activity of Wnt/SPAC, which regulates the phosphorylation state and activity of NCC. So our hypothesis in terms of KIR4151 being a drug target is that an inhibitor will lower what calls membrane depolarization, decrease intracellular chloride, which will inhibit Wnt/SPAC kinases and dephosphorylate NCC and reduce the activity. This could be useful in I think, well, let me just back up and say that in terms of target validation, we have nice genetic validation in a syndrome called East syndrome, which I'll talk more about in a minute. And we think that an inhibitor of KIR451 might be beneficial in a setting of loop diuretic resistance, where you have hypertrophy of the DCT and activity of NCC, but also in chronic kidney disease. And I can talk more about that later. We also think that an activator of KIR451 might be useful in terms of Gittleman syndrome, which is caused by loss of function mutations, hypofunction of NCC, and also E syndrome, which I'll talk more about. And there's actually some very recent pharmacological validation where it was shown that calcineurin inhibitors, which are often used in kidney transplantation, actually increases the activity of KIR451 via phosphorylation-dependent mechanism, and actually does expand blood volume and pressure, as you would predict from this model. The problem with testing these ideas, these hypotheses, is that there were no pharmacological tools available to really rigorously test this hypothesis, de-risk those targets. And so that's where we came in. We performed a high-throughput screen of about 80,000 compounds at a concentration of 10 micromolar in 384 oil plates using a thallium flux assay, which we heard about yesterday. You can see in the scatter plot, we have DMSO and fluoxetine, which is a control inhibitor, which inhibits most KIR channels, and we use this oftentimes in a screening campaign. Here we're looking at percent inhibition of our DMSO-treated wells in a 384 oil plate with fluoxetine. We found lots of interesting small molecules, mostly inhibitors. So this is a sort of a scatter plot. We found lots of inhibitors. We also found some really nice activators. I'm highlighting one of the hits from the screen, which is shown here. It's a compound that we call VU690. I'm not going to go into the story there. I want to get on to the unpublished data. Initially, we found it to be an IC50 about 1.5 micromolar. We generated maybe 50 analogs. We're able to get that potency up to 240 nanomolar. We're currently further developing that with a new grant, trying to get the potency further improved and make it useful for doing in vivo pharmacology. I'll just point out that VU720, which is this optimized compound, is highly selective for KIR451 over 12 other endorectifiers that we've evaluated. This is just some patch clamp data, cell-attached patch from CHO cells, showing that 10 micromolar VU720 does indeed inhibit channel activity. So, my training and interests are in ion channel electrophysiology, physiology, and pharmacology, so we want to understand the mechanism of action. We typically do an experiment called a high potassium knockoff experiment, where we ask the question, does increasing the electrochemical driving force for potassium change the potency of the inhibitor for the channel? So this is just a concentration response curve of KIR451 currents, heterologously expressed in hex cells in this case. And we show that by increasing extracellular potassium, we get this large rightward shift loss of potency, which is consistent with this model, where VU720 binds somewhere in the pore, and we're able to actually displace that with inwardly directed potassium ions. So it suggested that the VU720 binding site was somewhere in the pore. Now, because we've had lots of experience with other modulators of KIR channels, we suspected that the binding site might be near the so-called rectification controller. The rectification controller, turns out, is a common binding site for lots of the small molecules that we've developed. So I think it's fascinating that small molecules are taking advantage of these small molecule binding sites that have evolved over millions of years. So here's an alignment of that region between KIR4 and 51. In KIR451, we have a glutamate residue. In 51, we have an asparagine residue. So we hypothesize that mutation of that asparagine residue to a glutamate would lead to a loss of potency of the channel toward VU720. Now, this experiment came with the caveat that we're dealing with a heteromeric channel, and so we were aware of the possibility that making this mutation might lead to the formation of primarily homomeric KIR451 channels, which you can see down here in the CRC, is largely insensitive to VU720. So we took advantage of the characteristic unitary conductances of the heteromeric channel versus the homomeric channel, and that 5.1 has a higher unitary conductance than 4.1. And we showed that introducing this mutation didn't affect assembly, and we still get a heteromeric channel with the characteristic unitary conductance. And when we did the experiment, we saw that indeed mutation of asparagine 161 to a glutamate leads to largely a complete loss of channel sensitivity to the blocker. So the binding site, like we found with other small molecules, is at that rectification controller. But what about those activators, right? So these were the first-in-class activators of KIR4 and 5.1. Generally, across the channel family, there aren't a lot of activators, with the exception of activators of KATP channels, which are heterooctameric complexes, where a lot of the pharmacology is directed toward the sulfonylurea receptor. But in terms of just sort of apo-KIR channels, there aren't a lot of activators, again, with the exception of KIR3s. So we were really interested in understanding more about these activators. So this is the first one. You guys are the first ones to see the structure of a compound we call VU206, which is shown here. And this are just voltage clamp experiments in heterologously expressed 4 and 5.1 channels, showing that 10 micromolar VU06 has a dramatic increase, a dramatic activatory effect on the channel. This is just a current voltage relationship showing that it doesn't simply cause it to become leaky or something. You actually get a hyperpolarization consistent with channel activation. It's actually a nice pharmacological tool to work with. It doesn't take very long to activate. You usually get full activation within a minute. And this is just a partial list of the channels we've looked at in terms of selectivity using electrophysiology. And as you can see, it's not a great activator, about 11 micromolar, so we have lots of room for improvement, but no activity on 4.1, 4.2, or KIR4.2, which are also expressed in the kidney. So, bona fide activator that's selective for KIR4 and 5.1. Again, these are just single channel measurements showing that you get primarily an increase in the unitary conductance and the number of open channels, with, at least with this data set, not a significant effect on the open probability of the channel. So one of the first questions we asked was whether it was acting sort of at the PIP2 binding site or as a PIP2 mimetic, because it's well known that all KIR channels require membrane PIP2 for activity. And so to address this, we used a voltage-sensitive phosphatase, which is summarized here in this cartoon diagram. The VSP is basically the voltage sensing domain from a potassium channel fused to a phosphatase, whereby upon membrane depolarization, you get activation of the phosphatase, and this rapidly cleaves PIP2 from the membrane, depleting it and leading to channel inhibition. And you can see, if you look carefully enough, that activation of VSP actually leads to a rapid inhibition. Within 10 seconds, you fully inhibited the activity of KIR4 and 5.1. So what we've shown here is basically pre-depletion of the membrane PIP2 leads to the inability of VO206 to activate the channel, right? So it's not acting simply as a surrogate for PIP2. If you deplete PIP2, no activity of VO206. And we did the other experiment where we pre-activated with VU206 and then asked whether or not depletion in the presence of 206 would lead to channel inhibition. In fact, we do see that. So activation by 206, then activation of VSP leads to the rapid inhibition of the channel. So it doesn't appear to be working through a PIP2 dependent mechanism. So I mentioned East syndrome earlier. So this is an autosomal recessive disease. It's rare, less than 1 million people worldwide. It's caused by loss of function mutations in KIR4.1. It stands for epilepsy, ataxia, sensory neural deafness, and tubulopathy. So the epilepsy, ataxia, and sensory neural deafness are caused by loss of function of 4.1 in the central nervous system. The tubulopathy is associated with loss of KIR4 and 5.1 activity in the kidney. There have been at least 14 mutations identified to date. These are mapped onto a cryo-EM structure of KIR4 and 5.1 that I'll talk more about in a minute. And I'll just point out that the most common one is something called R65Q, or R65C is another common mutation. So we asked whether or not this activator was able to rescue the function of some of these East mutations. And so we did 2 experiments. The first was a thallium flux assay that I mentioned earlier, where we heterologously expressed either nothing the wild type channel or these channels carrying the indicated mutations. And interestingly, we find that VO206 leads to robust activation of the wild type channel as expected, and as well as a subset of these EAST mutations. Now, when you're doing these assays, these are 384 plates. You can have Ns for a single mutation of 20-something, right? So we wanted to make sure that we weren't biasing our conclusions toward having a high replicate number. So we did wholesale patch clamp experiments where we expressed the same mutations, and indeed we were able to see robust rescue in terms of the membrane potential of many of these, including the most common R65C. So this is the first sort of hint that some EAST mutations might be rescuable with an activator of the channel. So again, we're interested in mechanism of action, and for this, I worked with a great collaborator at Vanderbilt University, Erkin Karakis, where he worked toward developing cryome structures of the channel in complex with VO206. He used a concatemer of the channel where he connects KIR-401 to 5-1 using this linker. He gets beautiful size exclusion chromatography data. You see a single band there. I'm not going to bore you with all of the image processing, but we are currently at least at the stage of this image that we produce for a grant application. We're down to about 3 to 3.5 angstroms. And so here's the cryostructure. So you have a typical structure for an interrectifier, transmembrane domains, large cytoplasmic domain, and I'll point out that this is in the closed state, so these particles were generated with just detergent. We're now trying to generate, or we're making great progress toward generating structures in nanodiscs where we can add, for example, PIP2 to hopefully open the channel. But what we see is that there are actually 2 VO206 molecules per tetramer, and these are shown here in red. You can see in this close-up, they are situated between the pore helix and the helix bundle crossing here. VO206 actually extends toward the pore. This is the top-down view. Here's a potassium ion in the pore. And you can see it's wedged between 4-1-5-1, not between 5-1-4-1, interestingly. And it looks like it points an oxygen residue toward the pore, which I think raises some interesting questions about ion coordination in the pore. And I'm not going to show the data, but we see a minor sort of dilatory effect on pore diameter. This is just a close-up showing some of the residues that appear to make contact with VO206. Most of the interactions take place in the KIR-5-1 subunit, which makes sense if you're talking about a KIR-5-1 selective modulator. Interestingly, of course, we found that asparagine at 161 appears to be playing a role. And so we've just started the mutagenesis experiments to test this, but these are just some data that's hot off the press, where we mutated KIR-5-1 N161 to E like we did previously, and show that we get complete loss of activation of the channel with N61, or with the N161 mutation. And so to sort of summarize, we've developed some of the first selective inhibitors and activators of KIR-4 and 5-1. We are hoping to get a cryo-EM structure of the inhibitor, because I think it's fascinating that they can bind very closely in the pore but have completely opposite effects on channel activity. We think it's probably related to the fact that the inhibitor occludes the pore, whereas the activator sort of stands back and does something to improve conduction through the pore. We're continuing our binding site model with mutagenesis. We're also, and we usually take just an empirical med-chem approach. That's what our med-chem team at the Warren Center likes to do, just use the chemistry and SAR to drive additional rounds of chemistry. But this time we've started working with a new recruit to Vanderbilt who does computational docking experiments, among other things. And one thing that Paula did is she docked VO206 into the binding site, and it turns out, we learned, that the VO206 compound that we get is a racemic mixture. So there are R and S enantiomers. And if she docks the R or S into the binding site, she actually gets tighter binding with the with the S form of the molecule. And so we're currently having R and S separately synthesized at the Warren Center, and we'll test whether or not we get some bump in potency when using that enantiomer. We're also hoping, like I mentioned, to get a structure of the open state with nanodisk, where we can populate the nanodisk with PIP2, hopefully get the open state, because one thing we'd really like to understand is what are the structural basis of these loss of function mutations in E syndrome, and then how does VO206 reverse and rescue that channel function. And eventually, once we get these compounds optimized and in terms of their potency and DMPK properties, go ex vivo and in vivo to test whether or not they have the desired effects on renal function. And so with that, I will close. I'll thank Samantha McClanahan, who did a lot of the work that we talked about today, Eric Caracas and his postdoc hero, folks in the screening center, chemistry. Craig Lindsley is a longtime collaborator of mine, and he's involved in this project as well. Chong Ho is sort of the lead chemist on this project. Sasha and Elena did the single channel work for us, and of course, funding from the NIH. Thank you.